Okay, everyone. Hi and welcome. So today we are here in the second session of the Intro to Omics workshop. So we have about 30 people here in person and I guess 45 on Zoom. So hello and welcome to all of you. Very exciting. Just a quick announcement before we get started. We are going to send some emails soon about instructions for the code vignettes in the later examples. So keep an eye out for that. We are gonna definitely hopefully get that all working and ready to go soon. So we'll be able to let you know how to access those things. So for today though, we have a very exciting session number 2 of the Omics workshop, led by Dr. Melissa Fazzari. So Melissa Fazzari is an associate professor in the Department of Epidemiology and Population Health at Einstein, as well as Associate Director of the Biostats, Epidemiology, and Research Design Core, which is also called the BIRD Core. Dr. Fazzari originally joined Einstein in 2001 as a biostatistician for the Cancer Center. That volume. She received her PhD in statistics in 2007 after several years after several years at Stony Brook University and as the director of biostats for Winthrop, she rejoined Einstein again as associate professor in 2018. Her research interests include using machine learning approaches for subgroup discovery in clinical trials and predictive modeling. So Dr. Fazzari, please take it away, and thank you again for joining us.
Oh, thank you for having me, and I hope this session is interesting. So let me share the screen. Okay, everyone can see this, I'm assuming. So welcome to a crash course in biostatistics. What does this session cover? Well, we're going to explore a lot of the statistical concepts and methods that are really the underpinning of many of the packages and methods used in the analysis of omics data. So the goal here in an hour and a half is not for you to walk out saying, wow, I understand how to analyze every type of omics data and use all of these different packages. That's not going to happen today. The goal today is to gain a general intuition for the biostatistics concepts. And hopefully with this intuition, when you are attending the subsequent sessions, you'll be able to really understand much more about the methods going into these different packages. And so this session is not in any way a step-by-step tutorial for a specific package or a specific omic data type. That's going to be in later sessions where you're going to get that more specific instruction. And I should also say that I am in no way an expert at omics data analysis. I am a biostatistician, and so this talk is going to be really focused on the biostatistics. However, all of the examples are going to be omics data examples. So with all those disclaimers in mind, I'm going to give you the outline of my session. So how this outline came to be is that Alex sent me a list of all of the different topics and what statistics people are planning to present or use in their different sessions. And so that's how I decided what we needed to discuss in this kind of crash course. So here's the outline. You can see that we're talking about a lot of things in an hour and a half. I try to follow kind of a nice intuitive path, but by the time we get down to item 6, we're going to bounce around a little bit. And it's only because I want to touch on some topics that are going to be covered in later sessions that may not necessarily flow quite so well. So forgive me for that, but I think if I can give you sort of that brief introduction to all of these different concepts that, you know, hopefully in the next sessions, you'll feel much more confident about the biostatistics. Okay, so with that in mind, let's start with the basics, probability distributions. And what is a probability? And I appreciate this might be too easy for some people. or it might be just right for other people. Understand we have a very heterogeneous group here, so we're going to start with some of the basics. A probability distribution is a mathematical function that assigns probabilities to different outcomes. So every probability distribution is characterized by a set of parameters. So for example, if I tell you I have a normal distribution, it's going to be defined by two parameters, the mu, or the mean, and the sigma square, the variance. Okay, and we can see a few examples of normal distributions down here. These all have the same mu parameter, which is 0, which is where the distribution is centered. And they only differ because this sigma square value differs. Okay, and so this black line, this distribution is governed by a variance that's smaller than this purple line. So that variance really is controlling the spread or the fatness of that characteristic bell-shaped curve that we know with a normal distribution. Okay, so suppose we have gene expression measured for a specific gene. Again, this is a very simple example, but one gene, 25 patients, we randomly sample from the population of interest. Okay, so we can, in our random sample of 25 patients, compute a mean, so say it's 22, and a variance. And this is what we call descriptive statistics. And we could even assume maybe that these statistics of the mean and the variance are reasonably close to what's the true parameter values in the population. Because remember, when we're doing this kind of approach, we are assuming that there is some population and these gene expression levels are being generated by a process that's governed by a true mean and a true variance. Now, so we get our mean and variance of our sample. We can assume that maybe they're close to the true parameter values. However, there's a lot of uncertainty with this, right? Because we only have a small sample. We have a sample of 25 people. And we know that if we took a brand new sample of patients from this population, more than likely we would get a very different mean and a different variance estimate, right? So ultimately, what we want to do is use our data set of 25 patients to draw conclusions about what the gene expression levels are in the target population, what these true parameter values are. So we want to perform what's called statistical inference. Okay, so if we can assume that our data were generated through some process with a specific probability distribution, then we can quantify uncertainty. We can quantify probabilities, right? So we can, for example, look at state the probability of observing an average expression level of 22 or more, given that in the population, this gene is not expressed. So if we assume that the gene isn't expressed, and that's the truth in our population, that's the truth, what's the probability of observing an average expression level of 22? We can also use probability distributions to compute confidence intervals and p-values and simulate data even from this known distribution, assuming various conditions for power calculations, right? So probability distributions are really, really vital for statistical inference. So the normal distribution is probably the most well-known distribution, but that's of course not the only distribution. So there really are two main branches of distributions. We have the discrete distributions and the continuous. Now the discrete are exactly how they sound. The variables can only take a finite number of values. So counting variables are a perfect example of that, right? You can have 0, 1, 2 as a count. You can't have a count of 4.3, right? This is a discrete variable. And we have three discrete distributions that we're going to talk about today: the binomial, the Poisson, and the negative binomial. Now, all of these are very common in sequencing data, right? So these probably are familiar to a lot of you. Now, with each distribution, I've given the parameters. So with the binomial distribution is governed by n, which is the number of trials, and p, which is a probability. The Poisson is governed by this one mean parameter. And then the negative binomial is governed by two parameters, the mean and this dispersion parameter. And we're going to talk about all three of these in more detail in a moment. Then we have continuous variables, which these distributions, they can take any value within an interval. So an example would be body weight, right? You can have a weight of 64.3 pounds, things like that. And the most common distribution for a continuous variable, of course, is the normal distribution. And the normal, like we talked about, is governed by these two parameters, mu and sigma square. And then we have these others that I've kind of put an asterisk next to because they are characterized not by parameters, but by things called degrees of freedom. And these three distributions are normally used when we are comparing a test statistic to a distribution. And we'll talk about those a little bit. All right, so we're going to go through some of these distributions very quickly, just so everyone is on the same page. Binomial distribution. So the binomial distribution is a sum of independent Bernoulli trials. So what's an easy example? You have a deck of cards. What's the probability of getting an ace? If you keep putting the card back and you have 10 trials, how many times are you going to get an ace out of 52, right? So you have 10 trials, each has a common probability of event P, which in this case for an ace would be 4 out of 52. And so we can say that the number of aces being pulled out of a deck of cards in 10 trials, that's our Y variable, that's our outcome. This tilde denotes that it's distributed as, and then here we're saying it's a binomial random variable, and it's governed by N and P. N are the number of trials, and P is the probability. And so the mean of this distribution is N times P, and the variance is NP times 1 minus P. And I should say that, a Bernoulli trial, N is 1 here, okay? So an example of this, 20 patients in a study with a probability of a side effect of 5% for each patient. We're assuming that each patient is independent. And by independent, I mean that if someone has a side effect, it doesn't influence anyone else's probability of having a side effect. So the probability is 5% for each patient. So we can assume that it's distributed as a binomial random variable. The number of side effects with a N of 20 trials, 20 patients, and 5% probability. And so knowing this, we know that, we expect one patient to have a side effect, right? Have some sort of side effect, because we know that the mean is 20 times 5%, and with variance of 0.95. And this is what the distribution could look like. All right, so a special case of the binomial distribution is when N is large and P is small. Okay, and this is a Poisson distribution. Okay, and Y is the number of independent events that occur in some sort of fixed interval of time or space. And so again, Y is distributed as Poisson with a mean of lambda. And the property of this distribution is that this one parameter not only describes the mean, but it also describes the variance, okay? And this is actually, quite a restrictive assumption. It's saying that, your variance is completely determined by the mean. And this may or may not hold. And we'll see this later when we're talking about RNA-seq data. But this is sort of what the Poisson distribution looks like with certain parameters, with a certain mean parameter. This one is 4. So you can see that it's a discrete random variable. You can only take whole number values. There's quite a few zeros. So this is Poisson. And then finally, of the discrete distributions, we have the negative binomial distribution. And so this is governed by 2 parameters. It's governed by the mean parameter and then this dispersion parameter. Like I told you before with Poisson, that's kind of a restrictive assumption, right? That the mean is equal to the variance. So the negative binomial distribution relaxes that assumption and says, okay, you know what? We're not going to make that assumption about the variance. We're going to allow the variance to be a quadratic function of the mean. Okay, so the first term here represents the Poisson variance, And then the second term represents this extra variation. Okay, and this negative binomial distribution is really an important distribution in RNA-seq data. And you see it in almost every package, I think. All right, and then finally, moving on to the continuous distributions, we have the normal distribution, which everyone probably knows about. It's our, bell-shaped symmetric distribution. It's governed by the mu and the sigma squared, the mean and the variance. Unlike the Poisson and the negative binomial and the binomial, this mean and this variance, they are independent. Okay, they are not functions of each other. So that's a difference. And also we can see that we have continuous values here, not counts. And so an example of this would be gene expression, like a microarray, a gene expression level. We see and use the normal distribution quite a bit. Okay, so how do we know which distribution gave rise to our particular data, right? Because I just told you that we use these probability distributions to make inferences. And so valid inferences, of course, require an accurate statistical model. So if I have count data or ordinal data that I'm going to just, I assume it's normally distributed, that's not so accurate, right? And so the inferences made assuming normality you probably are not going to be that great. So how do you know which kind of distribution to use for your data? Well, first of all, look at the type of data you're collecting, right? So some clear giveaways are if you have count data. Do you have a lot of zeros? Do you have a big skew to your data? Are your data continuous? Are there some thresholds in your data set? So your data can give you a lot of hints, as can plotting, right? Histograms, QQ plots, all of these things can kind of tell you which way to sort of lean to in terms of what type of distribution to assume. And then, of course, previous studies and subject matter knowledge, because a lot of times if you have small sample sizes, any plot you generate, you can't tell anything from. And so you have to rely a little bit more on subject matter knowledge and the type of data you're collecting. All right, so I want to do a little detour before we talk about testing for differences between groups. And I want to talk about the unit of analysis, because this is really important, and it's important with a lot of omics data types. So what is the unit of analysis? Well, the unit could be anything, right? It could be people, mice, cells. It's important to understand that statistical tests and models, unless they are specifically for clustered data, assume that each unit of analysis is an independent sample from the population of interest. Okay, so, and again, independent means that measured value doesn't depend on any other measured value. So knowing my gene expression tells you nothing about your gene expression. Those are independent samples. Okay, so what is a technical replicate? Technical replicates are repeated measures from the same biological sample, okay? And these samples may be independent within that biological sample, but they are only independent measures of technical variation, measures of noise, measures of the equipment, the methods, the protocol variations, okay? And so the population with the technical replicate that you're sampling from is that specific biological sample. So if I have my gene expression measured 1,000 times, you cannot infer beyond me. Okay, that's the technical replicate. Now, in contrast, biological replicates are independent measurements from biologically distinct samples from the population of interest. Okay, so they are going to show both technical and biologic variation in their measurements. So the gene expression measurements that I get from myself and then from somebody else are going to show the technical issues and also the biological variation, and that I'm a different person than you are. Okay, and so it is the biological variation that allows you to generalize your experimental result back to the broader population from which you sampled. Okay. So common pitfall that we see all the time in design and analysis is treating technical replicates as independent biological variation. And this has two effects. It first artificially increases the sample size, and it also reduces the measure of standard error, standard deviation. Okay, it's basically you're treating technical replicates as independent, but they're related because they're from the same unit, and you are underestimating the variability in your data set. Okay, so increasing sample size, underestimating variability means that you're going to get a higher chance of false positives. So let's look at an example, and this is a really nice paper that discusses the issue of technical versus biological replicates. So in this example, the authors of a paper were interested to see whether the expression of three proteins, A, B, and C, were altered in a knockout mouse model of a gene encoding protein X. So here is the graph that was presented. It looks great. We have this bar chart, which I actually despise, but we have a bar chart. The level is the fold change. We have some sort of standard deviation bar. We have an asterisk saying, yes, this is statistically significant. So it looks good, right? Protein expression looks to be increased for all three experiments, A, B, and C. But let's look at this now in terms of where these numbers came from. Okay, let's look at it in terms of the replicates. So in this first experiment, experiment 1, we have 5 replicates from one mouse, 1 from another mouse, and 1 from one more mouse. So we have 3 independent biological variants, right? What's the problem here? Well, we first of all do not have a sample size of 7, right? We really have a sample size of 3. And second of all, if we took the average of these biological replicates, we would not have statistical significance anymore. So treating these as independent, we have statistical significance, we have a p-value less than 0.05. But if we did the proper thing, which likely is averaging across these technical replicates to end up with 1 observation per mouse, we are no longer going to have statistical significance. So already for experiment A, protein A, we know that this is invalid, this is incorrect, okay? And most of the variation here is in this one mouse, and you know, it's incorrect. Because if you average this, there's actually a lot of variation between mouse 1, mouse 2, and mouse 3. So what about the second experiment? Well, that's even more problematic. First, we only have 2 independent observations, right? We have mouse 1 and mouse 2. Second, we have this kind of weirdness where for this first mouse, you have a very big variation in the fold change being computed. And so this makes you wonder what's going on with one or both of these technical replicates. And then, of course, because we only have 2 observations, we really shouldn't be computing any sort of test statistic at all. So this is problematic, experiment 2. Experiment 3 is the only one that is valid, okay? And this is because we actually have 7 biological replicates. And so the t-test and the p-value are actually valid. Okay, so again, we want to be sure what level of variation we're talking about. Are we talking about technical variation or biological variation? Okay, so inferences can only be made about the population from which the independent samples were drawn, not the technical replicates. So if you have 5 replicates from a single mouse or a single cell line, this is still very important information, right? It provides really an internal quality check, but you can only generalize to that one mouse or that single cell line. Okay. By the way, I can't see the chat unless I really go to, oh, feel free to write any questions. But yeah, so if someone wants to yell out a question, feel free, because I'm not going to be checking the chat. So now we're going to move on. Now that we have a sense of probability distribution and what kind of variation we have, what kind of replicate we're looking at, now let's talk about testing for differences between groups. Okay, so let's suppose we have gene expression levels measured for a particular gene. Again, just one gene for now, under two treatments, treatment A and treatment B. And let's suppose I have 15 biological replicates per group. We would like to assess whether the average gene expression value is different for different treatments. So this is a very, very common type of problem that we encounter. So the form of the outcome variable, and I think you probably know this after the talk about probability distributions, but the form of the outcome variable is going to dictate what statistical tests should be used. So if you have lovely, continuous, normally distributed data, you're immediately going to look towards t-tests, analysis of variance, linear regression. I mean, of course, you can do a non-parametric test, but you would want to use the most powerful test possible for this continuous normally distributed data, which are going to be the t-test and analysis of variance type tests. Now, if you have markedly skewed data and you have a small sample size and it's non-normal and you know, but you cannot assume a distribution, you then want to look more towards non-parametric tests. And by non-parametric, I mean we're not assuming a distribution for the underlying you know, probability. We're not assuming a probability distribution. If you have count data, so something 0, 1, 2, 3, 4, or you have, you know, success failure, something like that, you're going to look towards either the non-parametric tests or generalized linear models, because with count data, we know we can't assume normally distributed. So we need to assume a distribution like Poisson or negative binomial. And then if you have categorical data, you're going to be looking towards chi-square tests and Fisher exact test and things like logistic regression. All right, so two sample t-test. And here we're going to assume equal variance. What are the assumptions of the test? We have two independent groups. So in my case, I have group A, treatment A, and treatment B. Each observation falls into one of the two groups. We have independent randomly sampled observations. I told you we have, I think, 15 biologic variates in each group, and they're independent, normally distributed, and both groups have the same variance. And that is an assumption for the equal variance t-test. We're going to change that in a little while. Okay, so I don't really want to get into computing the test statistic. That's not the goal here. But what I want to show you when we're looking at this test statistic is what it's all about. Okay, this is a ratio, this test statistic. The ratio is the difference in the average expression in group A versus group B. Okay, so this is kind of your signal, right? The mean differences. This is what you can compute even without inferential statistics. You can compute a mean difference, right? So that's the numerator. And then in the denominator, we have some measure of spread, some measure of variability. Okay, and we're using this test statistic to test a hypothesis. And that hypothesis is that the group means are the same. Okay, that in the population where I'm pulling my data from, group A will have the same average gene expression as group B. These mu's are equal. That is the null hypothesis versus the alternative that the means are different. Okay, and like I said, we have this signal, the average difference, or the difference between the average gene expression in the two groups, and we're dividing that by this kind of pooled standard deviation that's telling us how much noise we have within each group, how much those gene expressions vary around their mean in each of the two groups. Okay, so let's look to see what kind of test statistic, you know, what we can expect from the test statistic. Well, in this case, right, we have the samples in blue from group A and the samples in pink from group B. And we can see that there's very little true signal here, right? The averages are very close together, the average gene expression. It's kind of close together. So they're kind of sitting on top of each other, these two distributions, okay? In this case, we have low between group variation, right? This is the between group variation, the average difference between the measurements in group A and the average in group B. So low between group variation relative to the within, right? We have more noise within our groups than we have signal. And if that's the case, this test statistic is going to be quite small, okay? Now, contrast that to a situation where we have this very big difference between the averages, right? So big that relative to the variance, it's kind of overpowering everything, right? That these distributions almost have no overlap at all. I can draw a line here and completely distinguish between group A and group A and group B. Okay, in this case, we would have high group between group variation and lower within group variation. So relatively speaking, the between group variation is much larger. And so this test statistic is going to be very big. Okay, and so that's what we're capturing when we're doing the two sample t-test. Okay, what does this look like under the null distribution? Because we can compute this test statistic for our sample, but for inferential statistics, we have to compare it to something. So what we do is we say, okay, under the null of no group differences at all, this is what I think, this is what the probability distribution function would look like for a t statistic, assuming no group differences, and with this degrees of freedom. And these degrees of freedom depend on the sample sizes in group A and group B. Okay, and so we compare our result. Let's say we got a test statistic of 4. Okay, we're going to compare it to this distribution, and you can see that the probability of observing something as extreme as a test statistic of 4 is very low, right? This is area of high probability. This is area of low probability. And so observing something that's sitting out here is very, very unlikely under the null hypothesis. And so we're going to have a p-value that reflects that. We're going to have a very small p-value because it's saying basically that under the null hypothesis, seeing something that extreme, a test statistic of 4, it's really unlikely. Okay, and so with that low p-value, we can reject the null hypothesis and conclude the alternative hypothesis. Okay, so in the t-test, I told you that one of the assumptions is that the variances between with the within group variance is similar between the two groups, right? That's an assumption. If that can't hold, if that doesn't hold, if we see that the variance of group A is very different than the variance of group B, then we're going to want to modify our statistical tests to account for those differences and variances. And so how we can test for this is do an F-test looking at the ratio of the variances. And again, compare our computed F statistic to an F distribution and determine how likely it is that we would have seen a result like this or more extreme under the null hypothesis. And so you can perform this kind of F test, this folded F test, if you want to test for the differences in the two variances, the ratio of the two variances being not equal to 1. Okay, and this is just to give you an idea of what the F distribution looks like with different sample sizes. And so let's say you run this F test and you get a significant p-value and you conclude that you have different group variances. Well, you're going to want to adjust your original t-test. And this is where this Satterthwaite's approximation comes in. Okay, instead of using the degrees of freedom that I showed you before, the n1 plus n2 minus 2 degrees of freedom, we're now going to modify it. And you don't have to ever compute this. This is just computed automatically. You're going to modify it, and you're going to get a slightly small number of degrees of freedom compared to the t-test, which assumes equal variances. Okay, so something like this. You're going to have a degree of freedom of 28 in the original and degrees of freedom of 20 in the corrected one. And you can see that in terms of the test statistic and the distribution under the null, it's not that different. So the probabilities get a little bit changed, but typically not that much. Okay. I know I'm going through a lot of things very quickly. If you have questions, feel free to ask them. Otherwise, I'm just going to plow on. So analysis of variance. When we had two groups, we did a t-test, right? We're looking at the differences between the average gene expression in group A and group B. What if we have more than two groups? This is where we can use something called analysis of variance. So again, the assumption is Y is normally distributed. Again, we're assuming that the within group variance, so the spread of the values within each group, we're assuming to be similar. Like the t-test, we are isolating the between group differences from the within group variance. Okay, so this F statistic, just like the T statistic, in the numerator, we have between group variability, and in the denominator, we have within group variability, our estimates. Okay, and so again, if the between group variability is dominating over the within group variability, our F statistic is going to be very large. And if it's very large, we will be more likely to reject the null hypothesis that there's no differences between any of the groups. Okay, so let's look at this in a graph so we can understand. So here we now have three groups, and here is our gene expression. And this is our scatter of points in group A. I mean, really, they should be on one line, but I just did this because it looked a little nicer. So we can see that there's similar scatter around the means for each group, right? We don't have any big concerns about the variance being that different. But we have a group mean here for A, group mean for B, and a group mean for C. And so what analysis of variance is doing, it's partitioning the total variability in gene expression, right? So we have, first of all, this first component, which is the deviation of the group mean from an overall mean. Okay, so we have these three deviations, the three groups, how their means deviate from the overall mean. And then our second component is how each individual observation deviates from the group mean. Okay, so analysis of variance is partitioning that out, right? Because if we have a really strong signal, meaning these group means are very, very different, and we have very low variability around these group means, meaning that all of the different data points in group A are clustered tightly around their group mean. If we have that situation, we're going to have a highly statistically significant result. On the other hand, if we have differences in group means, but we have huge variation in the values within each group, so the spread, it's very, very widely spread data, very noisy data, we're not going to have statistical significance. Okay. So that's analysis of variance. So very similar to a t-test. Again, we are partitioning that variance into between group variance and within group variance. We can always represent an analysis of variance using a linear regression model. We can't go the other way always, but ANOVAs can always be represented using a linear regression model. And by linear regression model, I mean just this kind of model where we're relating the average value, the expected value of gene expression, or Y, as a linear function of different variables. And these parameters are the weights for each variable. So here I have a very simple setup. I only have my one variable x, and x is equal to 1 if you're in group A and 0 if you're in group B. And so the expected value of y is equal to beta naught if you're in group B, or it's equal to beta naught plus beta group A if you're in group A. Okay, so beta naught is the expected read count or gene expression or whatever you're measuring for group B samples. And the beta group A is the expected difference in read counts between group A and group B. Okay, and again, here we have a regression line. And this is a very simple example because I only have this one variable x, which is 0 or 1, which is 0 if it's group B and 1 if it's group A. And we see here the same situation like analysis of variance. We have the regression line, and the slope of this line is giving us the effect. It's telling us how gene expression is changing when you go from group B to group A. We have two situations here that I'm showing you. One is where we have a lot of error around the regression line, right? The residuals around the regression line are very spread out. And then we have a situation where we have less error around the regression line. Okay, in this case, this bottom figure is going to have a linear regression with a statistically significant value for the mean difference compared to this one. Okay, so we have a linear model. Now we're going to talk about generalized linear models, okay? Because the linear regression model, we have assumptions, right? We have normality of our data, and you know, we assume that the variance is roughly equal at every level. We sometimes need to generalize that, particularly as we move away from the normal distribution. Okay, so a generalization of the linear regression model, quite aptly, is called generalized linear model. Okay, and so it's still the same linear function of the variables. In this case, I have one variable, whether you're in group A or not. But now I have this more generalized term, this function of the expected value of y. Okay, and so the assumptions here are that the response variable has a distribution from an exponential family. So y could be binomial, it could have negative binomial, Poisson, or normal distribution. And then there's some transformation through this link function that relates the transformed value to a linear function of the x's, the x here. Okay, so let's look at them for different distributions. Okay, so a binary outcome variable, right? We have a binomial distribution. I have one here because it's one individual, and governed by the probability parameter. The link that we're going to use is a logit link. So we're going to take the log of the odds, p over 1 minus p. So when we do that, we end up with a logistic regression, right? And the effect being estimated is the difference in log odds of event, the log odds ratio, okay? And we normally, at the end, exponentiate our estimate to get the odds ratio. Okay, if we have a Poisson distribution, we are going to use a log link, and the effect that we're going to estimate is the difference in the expected log counts. And if we have negative binomials, similarly, we will use a log link and look at the difference in expected log counts again. And then, of course, if we have our normally distributed outcome, we can use what's called the identity link, right? We're not transforming this at all for linear regression. We are just directly relating the average to the average gene expression or whatever to the variables to our linear function. And so what we're getting here, the effect estimated, is the difference in expected means. Okay, so when someone says I'm using a generalized linear model, they can't just say that, right? You have to say with a link, with a log link and assuming this kind of distribution. All right, and we're going to see a lot of these kinds of models in a lot of the omics packages. And we'll discuss, we'll discuss right now. So here's an example using bulk RNA-seq data. So the read count here is a measure of the expression of a gene in a given sample. And so this is what your data look like. You have a bunch of independent biological replicates, you have a bunch of genes, and then you have the read counts. Okay, and so right away we know that we have discrete numbers, right? We know that we don't have counts of 3.2, and we can't have values less than 0. So right away, you should be thinking, okay, I have count data, Poisson, maybe negative binomial, okay, generalized linear model, or maybe Wilcoxon test, but let's talk about a generalized linear model here. Okay, so Just a bit of background for anyone in the audience who's not a genomics person. So RNA-seq gives you a snapshot of transcripts present in the sample. And so each sequence is considered to be binomial, have a binomial distribution with some probability P. And P for each gene, right? So sub K, so K is the gene. P equates to the probability that the sequence is aligning to gene K. And so this is a series of random events, a sum of a series of random events. And we can assume that they're all independent. And so we can see here, that we have, some level of alignment, and that's indicating how much is present in the sample, how many, what RNA transcripts are present in the sample. Okay. So because we have a lot of trials, and each one has a very small probability of success, we naturally look to modeling the data using Poisson distribution. Because like I told you, for that distribution, it's large N, small p, this counting data, we're looking for the number of successes. So it kind of fits the RNA-seq data with the Poisson distribution. And so what we're saying there is if the Poisson distribution applies, then every gene follows the Poisson distribution. And that lambda parameter is the mean number of transcripts for that gene. Okay, so what is understood in the RNA-seq community is that the Poisson assumptions hold really well for technical replicates, okay? But it does not hold so well for biological replicates. When you look at biological replicates, we are now combining the technical variability with true biological variability. What happens is the variance is now much larger typically than what is predicted by the Poisson. Because if you remember, that Poisson distribution has that very restrictive assumption that that lambda, the mean, is equal to the variance also. And so if we just blindly assume a Poisson distribution for RNA-seq data, we are ignoring this extra variation in the modeling. And so we can end up with a bunch of false positives. So, and here's a picture of this in case you couldn't imagine it. Here is where, to show you, here's where the mean count is higher. And you can see that the variance is higher than what we would expect. This red line is what we would expect under the Poisson distribution. And we can see we definitely don't have that. And so we need to think about modeling, using a distribution that is less restrictive. And so that is why we end up with the negative binomial distribution for RNA-seq data. So most popular packages are for RNA-seq data are based on this negative binomial distribution, okay? And they use a generalized linear model. And so let's go through this again. We are relating a log of the expected value, the expected read count. And so that's the log of that lambda. We are relating that to a linear function. In the simplest case, we have an intercept and just my group A indicator X, which is equal to 1 if you're in group A and 0 if you're not. In more complicated models, we can have a bunch of different variables if we have more than one group, or if we have other features that we want to adjust for, we can have more parameters. And then, so we have the link function, the log link, the linear function of the variables. And then we're making this assumption that the read count is distributed with a negative binomial distribution with lambda and alpha j, right? Because we're working now on multiple genes. So we'll index this with gene j. So here, and I know I said I wasn't going to do any sort of package or whatever, so I'm not even going to tell you what package this is from, but you all know what it's from. But here is an example output from the modeling. And so you can see that you have a bunch of genes, you have the mean of the normalized counts across all samples, and you get a log 2-fold change, right? That's that beta estimate, right? If we were comparing group A and group B, and then some measure of standard error, okay? And then you get your test statistic, okay? And this test statistic is just simply the ratio of the signal, which is the slope of that regression line, to the noise, which is the standard error of beta 1, which depends on the scatter of points around the line, like we saw in linear regression. And then we end up with some p-value. And so what this p-value is telling us, because it's so high, it's almost 1, it's 0.85, is it's saying that seeing a test statistic that's 0.18 standard deviations away from 0, right? Because if you look, this quantity is the number of standard deviations. To see something 0.18 standard deviations away from 0 is really not that unlikely under a null hypothesis. And so that's why your p-value is so high. Okay, so this is the kind of output that you're going to get. And again, this is based on that generalized linear model using a negative binomial distribution. Now, there is so much more to this than just what I said, because there are normalization issues, there are quality control issues, there's all different types of things. So I'm being very simplistic here, but this is the main part, the main thrust of the model is the negative binomial model. So if we had a huge data set, right, which a lot of statistics kind of assume that you have this like nice big data set and you can, your data can support anything that you're going to estimate. That's not the case with RNA-seq data. It's certainly not the case from what I've seen. So estimating the mean and the dispersion parameters, remember, we're doing this for every single gene, a different mean and a different dispersion parameter. It's really straightforward with a large sample size, with hundreds and hundreds of biological replicates. But for many RNA-seq experiments, you only get 2, 3, 5, 10, if you're lucky, biological replicates, which means that the estimates are really not that well supported by your data. And so they're going to be very, very unstable. And the issue with that is that we want to estimate these parameters properly because we want to see which genes are differentially expressed, right? And so if we overestimate dispersion, for example, we can really kill our power. And if we underestimate dispersion, we can have false positives. And so, there's an issue when you have small sample size. And so a lot of the packages, what they do is they allow for a common dispersion parameter to be estimated from the data. And then they shrink, and this is not something we're going to talk about today. This is something in subsequent sessions I am sure you will talk about, that you're going to shrink the estimates for dispersion towards this common dispersion parameter. So what that allows you to do is it says, okay, you know what? We don't have a lot of information from any one gene. So we don't have a lot of support for these models that we're building. And so if we borrow information across genes, so genes that have similar read counts, and if we borrow information, then we can use that to get a better estimate for the dispersion parameter. Okay, and so that's what a lot of packages do. And it helps with these kind of small sample sizes. And similarly, you can use methods that will shrink noisy estimates of fold change towards 0, right? So genes with low counts or high dispersion or few degrees of freedom, they have less information. They're going to be shrunk more towards a lower fold change estimate. So with small sample size, there are some tools available that can help you borrow information across all of the different genes that you're looking at and get better estimates for both dispersion and fold change. All right, what if you can't assume an underlying distribution at all for your data? So we just talked about, we have RNA-seq data, we assumed negative binomial, and we fit the model, everything is great. We have our genes, wonderful. What if you have data that you can't assume anything? You just don't have an underlying distribution that's reasonable. There are non-parametric statistical tests that you can use. So for two group case, we have the Wilcox and Mann-Whitney test. And this is a test used when you can't assume that you have normal data, or you don't want to assume negative binomial or anything like that. You want to use this kind of non-parametric test to test whether there are distributional differences between the two groups. There are fewer assumptions here. Of course, we still have the assumption that the observations from the groups are independent. And we also have an assumption that the responses are at least ordinal, which means that we can tell which of the two observations is greater. Okay, that's all we need to be able to do a Wilcoxon test. And sometimes this test, I mean, the good thing about it is that, we can run a test, we don't have distributional assumptions. The bad thing about it is that we lose power, right? Because we are taking our data and we are converting them to ranks. Okay, that's why it's called a rank sum. We're taking our data, we're making them into ranks. And so we lose information. But if you have really skewed data, this can sometimes be really helpful, because if you have a data point that's sitting way out there at 3,000 and your next data point is 200, if you order them, if you convert those to ranks, there's only one rank difference between those two values. And so it's kind of pulling in the outlier. And so that's a benefit to doing these kinds of non-parametric tests. So just very briefly, the general procedure, like I said, we convert all the data to ranks, and then we look at the sum of the ranks in group A and the sum of the ranks in group B, and then we compute a test statistic based on these sums. And then, like any other test statistic, we compute a probability of observing a value of our test statistic as or more extreme than the one we observed, right? This is the p-value. Through either a normal approximation or exact methods. And so this is another way we can test for group differerances. And of course, there are extensions to this. If you have more than two groups, you can use a Kruskal-Wallis test. There's a whole class of non-parametric tests that we don't have time to talk about, but I did want to mention it because these tests are regularly used in omics data analysis. Okay, I have a blank screen because I was told that it's probably a good idea to take a break. So do you want to take a 5-minute break?
Hi, Dr. Fazzari. Yeah, I think that's probably a good idea.
Okay,
Yeah, I think we can break for 5 minutes and come back to it. Thank you so much.
great.
If people have questions, please leave them in the chat, and we can take some time to read those. Thank you very much.
Should I share my screen again? I don't know if I can
All right. Uh,
while you're sharing.
yeah, hi, Dr. Fazzari. You can go ahead and share. We did
Okay.
have a couple of questions in the chat.
Yes, I saw them.
Um,
I looked.
okay, maybe we could tackle those before you continue. Is
Yes.
that okay with you?
Yeah, it's fine. Did I
All
do
right. Before
something?
we do that, I just want to say I think that this presentation is actually is really amazing. I have taken a look at some of the or most of the vignettes. And yeah, I honestly I can't say enough how relevant this is to the whole rest of the workshop. It is really perfect. It's a perfect intro. I imagine this is going to be the kind of thing that people want to return to a couple of times. Um, and yeah, these are really helpful slides and presentation here. So um, we had a first question here is could you give a more intuitive example or illustration of dispersion? So,
Yes. Okay. So think about it like this. If you have a gene with expression read count of 1,000 and a variance of 10, that is very different than a gene expression of 100 with a variance of 10 or a read count of 10 with a variance of 10, right? So even though it's the same variance, the variability of observations around the mean are going to be much, much greater than with the smaller read counts than with the larger read counts, right? Because if I'm telling you I have a variance of 10, and let's say, you have 1,000, so 1,010, 1,990, right? So relative to the mean, that's kind of a very small variation. When you have a read count of 10 and your variance is 10, 0, 20, right? So, I mean, it's a much bigger variability around the expected read count. And so you can kind of see it here, right? In these lower mean counts, you see a lot more spread to the data than you do up here. And so really, that's what I mean when I'm talking about dispersion. Does that answer the question? You think of it in
right.
terms of the coefficient of variation. Right? So the dispersion is related to the coefficient of variation, and coefficient of variation is the standard deviation divided by the mean.
Right. So high value there will indicate that the variance is a lot higher than you would expect.
Yeah. So
Well,
really we
that.
don't care so much about the variance, because that is so related to the average read count, right? And so we wouldn't want to do a test statistic based on that variance, because then what would we do? We would only have low read count being statistically significant because the variance would be so high. So what we really care about is that dispersion parameter when we are assessing which genes are differentially expressed. Okay, because that's really measuring that biological variability. And then well, I hope that, if it's not enough, I am sure that in the next session, not to push it off onto Kith, but I'm sure in the next session, you're going to get a lot more helpful, intuitive, data-driven examples of dispersion. And then the other question was concerning the log link. And so I just want to be clear, we're not log transforming the variables, right? We are using a log link. So that's different. But in terms of how we use this in reality, so if we had RNA-seq data, for example, let's go to the RNA-seq model, right? So we are assuming for our generalized linear model that this log of the expected value of the read counts is linearly related to our function of variables in the model. Okay, that's all we're assuming, is that this linearity, this linear function is related to the log of the length, not directly to the read count itself, the log transformed read count. Okay. And so that's the way you use it. It's different than just log transforming your data. Like let's say you have you want to use linear regression and you say, oh, okay, let me log transform to get it looking a little more normal, normally distributed. You log transform it and then you run a regression. That's different. That's a little bit different than what we're talking about here. So let me go to, is that okay?
Please continue.
Okay, so let me go to p-values quickly. And I'm realizing it's already 1:16 and I think I'm only halfway through. So I might have to either move quickly or cut some things. So what is a p-value? The p-value is the probability of observing a test statistic as or more extreme than the one you observed with your sample, given the null hypothesis is correct. So under the assumption the null hypothesis is true, what the p-value is telling you is the probability of seeing a test statistic as large as the one you observed, right? So anytime, like when we were doing the t-test or the ANOVA, we were comparing our computed test statistic from our data to this theoretical distribution under the null hypothesis, generated under the null hypothesis. And we were saying, okay, wow, our test statistic was 4. Chances of seeing a 4, given the null hypothesis is true, is very, very, very unlikely. And so our p-value is very small. Okay, so that's what a p-value is. Usually when we talk about statistical significance, we are talking about a p-value cutoff set at 0.05. For better or for worse, that's what we have all decided is statistically significant. But you think about what I just showed you with that RNA-seq data example. We have p-values for all the genes in our data set, right? We are doing the same generalized linear model for every single gene. And then we are taking the estimate for our slope and we are dividing it by our standard error. And we are generating a test statistic and a p-value for tens of thousands of genes, well, thousands and thousands of genes. Okay, so even if you focus, you say, okay, we have all these p-values being generated for the 20,000 tests that we've done. We're just going to cut off at 0.05. Well, the problem with that is that every gene has a 5% chance of being a false positive, right? Because the definition is that we're going to allow p-values less than 0.05 to be considered statistically significant, which gives a 5% chance under the null hypothesis of being a false positive. And so if we have 10,000 genes, that means we have 500 potential false positives, which isn't great, right? Because then who knows what you're identifying. So what people do sometimes, not very often, is a Bonferroni adjusted p-value, which is a really, really strict conservative way of adjusting for the fact that you've just tested 10,000 genes. And what it does is it divides the 0.05 threshold by the total number of tests, which is 10,000. So it now says, okay, fine. You want to do 10,000 tests? Your p-value to be statistically significant is now much, 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 much more rigorous, right? You have a much higher hurdle to jump through to declare yourself this particular gene statistically significant. Okay, this is very conservative and not used very often because if you use this, you end up normally not really ever having very many genes be declared statistically significant. So like we said, the raw p-values are too lenient, the Bonferroni is too strict. So let's now look at false discovery rate, the FDR. The definition of the FDR is the expected number of false positives over the number of significant genes. So it's the proportion of falsely discovered genes in our set of significant genes. Okay, so if our FDR is 0.05 for 100 of our identified genes, we estimate that there's no more than 5 falsely estimated. That's the false discovery rate. And with the FDR, we can compute a Q value. And the Q value is sort of the FDR analog of the P value. And so we have one FDR to define our set of significant genes. But we have a Q value, just like a P value, we have a Q value for each gene. But it tells us something a little bit different than the p-value. So let's look at one of these, this q value by story. So story defined the q value for a particular p-value as equal to this quantity. So it's the probability of rejecting the null when the null is true, right? This p-value. The probability of seeing, our test result as or more significant, as or more extreme than our test results. What's the probability under the null hypothesis? And then it's multiplied by the total number of genes, and then the proportion of null genes in the data set. And then it's divided by this differentially expressed set, all the p-values with p's less than or equal to whatever p-value you're at. And I realize this is very confusing, so I'm going to go right into an example. But what I want to talk about first is how we come up with this number of null genes in the data set, because we don't know that, right? We have 10,000 genes. We have no idea how many null ones we truly have. I mean, we can make a guess. We can say, yeah, probably most of them are noise. Most of them have no differences between treated and treatment A and treatment B. But we'd like to have a basis for which to estimate this proportion of null genes in our data set. So how do we do that? What we can do is look at p-values under the null hypothesis. And it turns out that p-values under the null hypothesis are distributed as a uniform random variable. So by uniform, it means sort of equally likely under all values from 0 to 1. And so we can use this information, right? So this is what we really have. We have a bunch of p-values from our experiment. Most of them are greater than 0.05. So signifying that most genes are uninformative. And then we have this kind of peak here. And so what we want to do is, of these, say, 500 genes that are identified with very high p-values, we want to separate out the true positives from this kind of background false positive, okay? And so that's what we're going to do if we assume this uniform distribution and we look at the p-values greater than 0.05, and we say, okay, because we have a uniform distribution and we know that this many genes have p-values greater than 0.05 in our data set, we're going to double that and use that as our estimate for how many total genes that are null in our data set. Okay, so we are going from here, counting up all of these genes, and then doubling it as a way to kind of draw this line. Okay, and so in doing that, in computing this proportion of truly null genes using that method, we can come up with a very reasonable estimate. Here it's 0.88, so 88% of the genes, or 8,800 null genes out of the 10,000, based on 2 times the number of genes with a p greater than 0.5. Okay, and so how does this impact your original p-value? Well, for your first gene, this is the original p-value. This is the story q-value. And so what this is telling us is that out of 8,800 truly null genes, we expect less than 1 gene to have a p-value of 0.00087. Here I'm talking about this bottom one, sorry. So out of the 8,800 truly null genes, we expect less than 1 gene to have a p-value more extreme than this. So we observed 6 genes, right? 1, 2, 3, 4, 5, 6, with such extreme p-values. And so the expected proportion of false positives is 12.8%. And I realize I'm rushing through this, and if I'll share my slides so you can kind of go through this yourself. But this is how the computation is done. Okay, so the p-value 0.0087, we expect a truly null gene to have a test result as a more extreme, very rarely under the null hypothesis, right? We compute this p-value without considering the number of genes being tested or the number that are significant. But with a Q value of 0.12, what we're saying is of the 6 discovered genes with Q values less than 0.0087, 12% are expected to be false positives, okay? And so that's the minimum FDR that you can call this gene statistically significant. And if we took that proportion, that 88.8%, and we made it 100%, so assumed all of them are truly null, then the Q value is obviously going to be higher. If we assume 50% are truly null, then the Q value is going to be much lower. Okay, so that's sort of the intuition behind p-values, Q values, and FDR. Okay, so here's where I'm going to start bouncing around a little bit. I wanted to cover principal components analysis because it's going to be touched on in a lot of the sessions, and certainly for packages for RNA-seq data, you see principal components analysis in the graphs a lot. So I wanted to give you some background on it in case you're not familiar with it. So let's suppose we have two variables, x and y. The variance of x is a representation. It can be represented by the spread of values along the x-axis, right? Variance, it's the spread of values. And similarly, y, it's the spread of values along the y-axis. Okay, but when x and y are correlated, right, we can't just characterize, like, so here's an example of a cloud of data points. You know, this could be whatever. This could be gene 1 and gene 2. This could be anything. And we have a cloud of data points, and we see that there's some relationship between x and y. We know x and y are correlated. So we can't just look along the x-axis and along the y-axis because we have this direction here. So how we can describe this data set is through not just the variances, but also the covariance, right? And the covariance means that as x is increasing, what is y doing, right? So the change in x and the change in y. And so the diagonal spread of the cloud of data points is described by this covariance term. Okay. So here's some examples of different covariance matrices. So here we have this positive covariance. Here we have a negative covariance, right? This is the direction when it's positive. This is direction when it's negative. Here we have independent, x and y are independent, and x has much more variability than y. And here's the opposite situation where, yes, they're still independent, but now y has a lot more variability than x. Okay, so this is just a few examples. Okay, so we want to find a vector that points in the direction of the greatest spread of the data. Okay, this is what principal components is trying to do. We are trying to find the direction where the data has the most spread. And so if we have independent x and y, there's no covariance, this is easy. We can just do a line along the x-axis and a line along the y-axis, right? And these are orthogonal, right? They're orthogonal because x and y are you know, well, that's how we're defining the principal components. Is, you know, the first principal component is going to capture the maximum spread in the data, and then the second principal component is going to be a vector orthogonal to the first one. But here, when we have two independent variables, the principal components align themselves perfectly with the x and y axis. Okay. So when we have a non-zero covariance, the principal component is now going to not align with either the x or the y-axis. It's instead going to kind of go through this cloud of points and try to account for the maximum variability. Okay, and that's what we're seeing here. Okay, and so when we perform a principal components analysis, we're trying to, you know, create these vectors that are accounting for as much of the variation in that cloud as possible. And so we can see that these principal components are linear functions of x and y. We're combining x and y in a way that this new function accounts for as much variance as possible. So let's look at it with RNA-seq data. And I love this. I love the internet, by the way. You can go online and you can type in PCA and, get these beautiful graphs. And this, by the way, if you go online, you can like spin it around and it's really very cool. I just took a screenshot of it, but there's so much online now. So this is what PCA is doing. If you look at this is now 3D, and you look to see the direction of maximum spread of the data. We have in this direction, and then we have in this direction, and then we have one projecting out. Okay, so if we take the first two principal components and plot the data, okay, so we are taking away one dimension of information, right? We're going from 3D to 2D, okay? And so we see the first principal component is in this direction, right? It's going through the cloud of points like this. And then the second is orthogonal to that direction, and it's going this way. So we can see that here's the data represented in 2 dimensions. So we've lost some information, but we've not lost that much because this third principal component really didn't have much variance associated with it. Okay, and so that is what, when you're talking about principal components and dimension reduction, this is what you're talking about. You're talking about going from multiple dimensions, here 3 dimensions, down to 2 dimensions. And trying to keep as much of the information in the data as possible. So if we looked at this for RNA-seq, now think about your 10,000 dimensional data cloud, right? All your different genes. It makes sense that if you want to take a step back and sort of get a global feel for what's going on in your data set, you might want to look at the first couple of principal components to see if, see the general pattern in the data. And so here we have the first principal component, which accounts for about 43% of the total variance, and the second principal component, which accounts for about 23% of the total variance. So it's a decent amount of the variance that's being accounted for in these 2 dimensions. And again, principal component 1 is a linear function of all of the genes. Principal component 2 is a linear function of all the genes. They are orthogonal. What can we learn from this? Well, if we look at the principal components and we plot the specific samples, so here we have 3 controls and 3 infected samples, we can see that, first of all, we have separation between the infected and the control, even in just this 2-dimensional space. So it's kind of giving us a sense that there is some sort of signal from the RNA-seq data that we're actually able to separate out the infected from the control. Okay, and now remember, PCA is only looking at gene expression or gene read counts. It is not looking at whether someone is a control or an infected. It's doing it without looking at that. And so, the fact that we're seeing this separation is telling us that, there might be some information. The other thing that this could be telling us is this control sample seems to be sitting out on its own, right? At least in this 2-dimensional space. It's kind of sitting away from the other control samples. So this is a way of allowing us to maybe isolate whether this sample had some technical issues, or maybe there's another variable that can explain why control 1 seems to be not clustering with the other controls. Maybe this is an older individual, maybe this is male and these are females, who knows? But it's allowing us to explore the data a little bit, and explore it in 1, 2, or 3 dimensions is much easier than trying to explore it in 10,000 dimensions. And so that's why we use principal components analysis. So let me see what time. Oh my goodness. Okay, I'm going to skip over gene list overlap. You can have my slides. Really, this is, when we're looking at 2 gene lists that have a set of statistically significant genes, how can we tell what the probability is that the overlap is meaningful versus just random? And we can do that with a Fisher's exact test. but I wanted to get a little bit into false discovery. We can come back to that if there's time, but I wanted to talk a little bit about false discovery and sample size computations. So there is a article talking about the why most published research findings are false. And I think everyone should read it at least once in their career, because I think it's really important to understand, how likely it is a false positive can happen in your research. So there's something called the winner's curse. So here I simulated 1,000 studies, and the true effect size is 0.3. Doesn't really matter what the effect size, what that means. It just means that here's the truth. The effect size is 0.3. If I sample 500, remember I'm simulating this so I can sample whatever I want. If I sample 500 per group, I'm of course going to have very, very high power to detect any sort of true effect, right? So I have very high power. The average p-value is very significant. Here's my average effect size. Okay, perfect, right? This is what I expect, right? I want to be able to precisely estimate what the true effect size is with a huge sample size. I can go down all the way to 5 per group, right? You have very low power, of course, because you don't have enough data to support, the model or whatever you're looking at, especially with an effect size of 0.3. So the average p-value, if significant, is 0.021, right? So most are not significant, but if they are, their average p-value is 0.021. But the average estimated effect size here is 1.04. And so what this is telling you is that with small sample sizes, if you happen to see a significant result, you are more than likely way overestimating your effect size. Okay. And the problem with this is, first of all, you publish this, oh my gosh, this is such an important gene or whatever. You publish it and you talk about the effect size 1.04. And then someone else comes along and they want to do their study and they look at your paper and they say, wow, okay, this had a big effect. I'm going to power my study assuming an average effect size of 1. Okay. And so what happens is the future studies get powered on an effect size that isn't really true. Okay. So you want to be aware of this, that small sample sizes can give you false signals, and effect sizes that are really just way, way, way overestimated. So I wanted to talk a little bit, this is the paper, that I mentioned, why most published research findings are false. What I want you to think about is this idea of prior probability of discovery. Okay, so suppose we have a study with 80% power to detect an effect of interest. Okay, let's assume the prior probability of being a true discovery, and this of course depends on what field you're in, let's assume it's 8%. Now this is probably much lower in a gene expression or RNA-seq study, right? For any individual gene, the probability of being a true discovery is probably nowhere near 8%, it's probably much smaller. So you have this study 80% power, you have this prior probability of being a true discovery of 8%. The probability of a true effect, given that it's given your discovery, is about 0.58. So the probability of a false discovery here is 0.42. All right, and this is something that, I didn't really think about until I read the paper. And then once I read it, it seemed so intuitive. Of course, right? If you have this low prior, very low probability of being a true discovery, and then you discover something, what's the probability of that actually being a true discovery? It's very small. And so keep that in mind when you're doing your studies and you're seeing this significant result, especially if it's kind of a surprise result. This wasn't an a priori hypothesis. You kind of scanned 10,000 genes and looked, and then you saw something. The chances of it being a true discovery are probably very, very small. And of course, that depends on this prior probability. And so But all is not lost. Yes, you have a higher chance of false discovery, but what's important to understand is the power of replication. And so if you look at one trial with this kind of dismal false positive probability of 42%, right? Let's say you replicate the trial, okay? 2 independent trials. Now look at the false positive probability. Okay, and this is close to what Fisher, who kind of invented the concept of p-value and this p-value threshold of 0.05, he viewed it as sort of a beginning, right? We tend to view a p-value less than 0.05 as the end, like, yay, we discovered something wonderful. But he viewed it as, this is a reason you should do a replicate study. Okay, a 0.05 is just a way to say, okay, yeah, do a second independent trial. And I think this kind of justifies that with a field, an area where there's a high level of a high likelihood of a null result, if you get a statistically significant result, think about the probability of a false positive, and think about how important it is to do that second and maybe even third trial if you really want to have a very low false positive probability. Okay, power and sample size related, right? So the power is the probability of getting a statistically significant test statistic, assuming there are true differences in gene expression or whatever. I'm sorry, I keep going towards gene expression, but it could be protein expression or methylation. So often we see sample sizes of 3, 5, maybe 8 with very little justification. Now, if you think about what we're doing with these models, especially think about the generalized linear model, we are fitting this model. We sometimes have variables that are beyond just the group variable. We're estimating the fold change, we're estimating the dispersion. These sample sizes, these small sample sizes are not likely to support that kind of model and testing very well. And so you really need to give thought to, your sample size computations. So how many biological replicates should you have? Well, as a statistician, I'm going to tell you as many as you can. Because why? Having many biological replicates allows you to estimate the variation for each gene, adjust for samples, adjust the model to account for confounders. You can look for outliers when you have more samples. And importantly, you can improve the precision of your estimates for gene expression and fold change and dispersion, all of that. So as I mentioned before, the commonly used packages have a way to kind of borrow information across genes. But you really do want to have as many biological replicates as you can, as is feasible, because you think about what kind of model you're supporting with your data. It's not nothing what you're doing. And so you want to make sure you can, have precise estimates. So power can mean many different things when you have 10,000 statistical tests, right? So with one test, we can define power as the probability. We reject the null hypothesis at whatever, probably 0.05 significance level, or an FDR of 0.05, given that the null is false, right? You assume some sort of alternative distribution and you compute power. And so with one test, you can define power like that. With multiple tests, power could have many, many different definitions, some of which I've listed below. The probability that every gene that we detect is truly different, the probability that at least one is truly different. And so there are many different ways to structure power in the multiple gene scenario. But we're not going through, how to compute power here. But what I do want to remind you of is that power is a demonstration under very specific assumptions, right? And so the more realistic you can make your assumptions, the more useful your power computations will be. Okay, so if you compute power, assuming a negative binomial distribution with a fixed mean and a dispersion, and 90% of the genes have fold changes of greater than 4, right? Wonderful. You're going to have a ton of power, and you're going to need 5 samples to have power. But the problem is that's so not realistic, right? That 90% of your genes would have huge fold changes. More realistically, you want to compute power assuming 10,000 genes with a a negative binomial distribution, and varying the means and dispersions with a tiny subset of differentially expressed genes with a fold change of greater than 2, right? You want power to be as realistic as it can be. And there are so many packages now that allow you to simulate power under various assumptions. And I would suggest when you're thinking about power and when you're, possibly presenting power estimates, do it in a table and vary those assumptions so you can see the differences, the changes in power based on the changes in your assumptions. Okay, so I've just pulled out one example package, this sample size, S size RNA. And this is the kind of stuff that they're going to ask you for, the number of genes, the proportion of truly null genes. You want this to be high, right? You don't want to assume that you have a lot of genes that are truly differentially expressed. You want a pretty low FDR value, either 0.05 or maybe 0.1 if you're, really in discovery mode. The minimum power should be 80%, 90% is always better. Some sort of average read count, some sort of dispersion, and some sort of estimate of effect for those differentially expressed genes. So you would need to have this in mind when you're computing power, all of these different things. And this is where you can kind of go in and play around and see what the impact is on power if you change the dispersion, for example, or if you change the fold change and see what happens to your power. In the time we have left, because I don't want to go too far, I wanted to promote my own department, my own division's thing, which is Birdhouse. We have a website now, and I have a link at the end of the talk, and you can see it right here, or you can just Google Einstein Birdhouse. And what this is, and it's growing all the time, but what this is a resource for you where you can, let's say you want to learn more about the 2-group t-test. You can go in and look at common statistical tests. There's for the 2 independent groups, the t-test, there's a basic overview. There's a description of the statistical tests used. And then there's our code to walk you through how to do this with your particular data set. And so, right now we have, it's not in its infancy, but it's still growing. So we're still adding things all the time. But I think you can definitely go there and find some really good tutorials and resources. We'll be adding much more about machine learning. There's not as much genomics on there yet, but there will be. And, a lot of these methods, as I've shown you today, because I'm not a genomics person, but I've shown you today that a lot of the statistical tests that we are presenting on Birdhouse, you're actually going to be using for these packages that you use to analyze omics data. So I would love if you went on Birdhouse and, of course, give your feedback and any suggestions. You can feel free to email me directly. In terms of other resources for analyzing omics data, because I just rushed through a lot of different topics and didn't cover any one in real depth. You have so many other resources online. edX courses and Coursera courses are such an excellent way of getting a good overview of the type of data analyses for omics data or for any data in general. There are different tutorials. This one I found particularly helpful. This is a paper discussing biological replicates. This is a DESeq2 paper, the original, and then of course Birdhouse. So there are a lot of resources out there, a lot of YouTube videos. So if you have questions or you want me to point you in the direction of anything, please feel free to email me directly. Okay, these are power calculation tools, and I know I went through them very quickly, or went through one very quickly, but here they are, and that's it. So, any questions?
All right. So we did have some questions in chat. Let me just make a couple of quick announcements and then we can go through that. So first, I just want to say, yeah, so as Dr. Fazzari presented, there's the birdhouse. We posted a link in the chat. The link to that is also shared on the Omics workshop website, which hopefully you all have seen. I really can't emphasize enough. It's a fantastic resource. And this is something that Dr. Fazzari and Dr. Mimi Kim have put together. And it's really amazing. And there's a lot of great tutorials on there with our code, as Dr. Fazzari mentioned. Additionally, please try to get on the HPC if you haven't done that before next week. And try to open up cluster.einsteinmed.edu. Some of the things that we talked about today will be demonstrated via code starting from the next session. Another announcement is that we were asked to mention that on May 14th, Dr. Fazzari is going to be giving an all-day R programming workshop along with some other biostats faculty. So that'll be on May 14th. So definitely please attend that. I think that'll be really fantastic. So let's get to our questions now. I think one question that was actually asked before that we didn't get a chance to get to was a question about sort of technical variance. This person asked, do you know if much consideration is needed for using methods for accounting for or removing technical variance, for example, in DESeq2, and can they be used the same way in all cases? So maybe a question about sort of like the concept of like regressing out or sort of dealing with technical variance in general. Do you have any sort of tips or any comments about that, Dr. Fazzari?
Well, my first comment will be tune in next week when the true experts in things like DEC2 are speaking, and I'm sure they will, tell you more information than I have about how to handle technical replicates in that specific context. My advice here, my general advice is to be aware when you have technical replicates versus biological replicates, and do not combine them. Do not treat technical replicates as biological variants. But I'll leave the more detailed information to the people presenting next week.
Okay, so tune in then. I think that is actually all the questions we had. I will just give a moment if people have more questions to type. If there's any questions in person, you guys can feel free to type them in the chat or just come back here. I guess that's probably the easiest way to do that. I actually had a question of my own while we're waiting, which is, I think a lot of people have this same sort of question, which is, how do you feel about the use of like cell cultures as replicates? You have like a cell line from, say, a tumor. You culture it, you grow it in 3 plates. Would you consider those as technical replicates or biological replicates, or is the answer that it depends, or?
I think probably I should say it depends. But if you asked me, I would say that's a technical replicate. To me, the biological variation comes from different cell lines. But I know a lot of people are going to strongly disagree with me on that. So I'll say it depends.
Okay, I do agree with you there. So we are waiting for other questions. If anyone has any other questions, we can go and ask. If not, I had one more on my own. So I think you may alluded to this before, Dr. Fazzari, but you mentioned a little bit about, okay, before I selfishly ask my own question, there was a question in the chat. So can different passage numbers be considered as biological replicates for cell lines? Okay, that's an interesting question. So say you have plated a cell line at passage 0 directly from the source tissue versus like from passage 10. Let me go ahead and say, I think that's probably experiment specific or tissue specific. but probably there's some strong biological changes between there. But did you have any comments about that, Dr. Fazzari?
I am not a genomics person. I'm not an expert on this, so I will defer to the experts. All I can say is that, you can only make inferences back to the population from which you sampled. And so if this is a single cell line, and you have done different experiments on that single cell line, to me, that's technical. I don't know the specifics of some of what's being asked, so I would hold that question for later sessions.
Yeah, I agree. That's a very like biology heavy question there. We had another question about what is the difference between p value adjusted, and the q value? Is there a difference, and when would you choose one over the other?
Yeah, so the p-adjusted, I mean, I don't know if that's a specific tool that you're using, a specific package, because adjusted p-value could mean anything. I'm not sure which one you're talking about, but do I have it really depends on what p-value adjustment method you're using. The thing to know about all these adjustment methods is that the ranked list is invariant to whatever adjustment methods you use, right? So if you were really in doubt, you could just take the top 10 genes or the top 50 genes, right? The ranks are not going to change no matter how you adjust the p-values. Using a p-value or a q-value or FDR just gives you a little bit of comfort to say, well, okay, with this gene list, this significant gene list of the top 10 genes, my FDR is 0.05, right? But it really doesn't change what you're doing. So in a lot of these experiments, especially when you're trying to discover relationships, you really want to look at the best candidates, right? And you will take a set of genes that are differentially expressed, and you will study them further. And so we don't necessarily need to have all of this, declaration of statistically significant or not. You can just take the best candidates and explore them. So yeah, I don't know which one is I don't think there is a best one to use. I just think if you compute them all and you're comfortable with what they're saying, in terms of false discovery, then you're going to take your best candidates no matter what.
All right, cool. So just a quick time check here. It is 1:58, so I think we should call it here. We did have a question about sort of recorded Zoom sessions and things like that. So just to say that we will be hopefully sending those out soon. Yeah, and again, just want to make sure people see that on our website will be all the details and information about the later sessions, locations, and that kind of thing. Most of the later sessions will be all in the Belfer building, in the Rickless Auditorium, which is in the Belfer lower levels. So yeah, with that, let's just give a quick round of applause to Dr. Fazzari. If you're on Zoom, please clap as well, okay? So thank you very much, Dr. Fazzari, for this, and we really appreciate it. And yeah, this will be very applicable for all the later sessions. So yeah, really, really want to thank you for that.
Oh, you're welcome, and I hope so.
Thank you so much, and thank you all for your interest and for attending.
Bye, everyone.
Thank you again. Dr. Fazzari, we will send you this recording as soon as we can. And
yeah, because I'm thinking we will put it on Birdhouse.
That'll be amazing. We do have a link to there on our website, so yeah, feel free to do that, and we will send that to you as soon as we can. Thank you again very much.
Okay, wonderful. Yeah, you're welcome.